So, uh, starting with like genome assembly construction and annotations. So, I just given a title here like process of putting new nucleotides into the correct order to get maximum information. So I hope everyone are aware about the what is the nucleotides and how the DNA is forming and how it's happening. So now coming taking that picture into the mind, the process of putting nucleotides into the correct order. So that is the thing, because we will get the sequences from the different technology and how it is putting into the correct order and getting the maximum information means we are generating the chromosomes, or you can say the contigs, and then getting the information from them. The information from there we are getting like non-coding RNA, non-coding things, non-coding part of the genome as well as the coding part of the genome. The process of putting correct order is very difficult. Difficult means it's like you can say there are millions of nucleotides are there for a genome, and we have to put it into the correct order. So that processes will take large time, as well as like you can say huge time, as well as that huge computational power, and the algorithm behind it very typical. You can say the algorithm are very much complicated over there. It depends on again the what kind of the data is there. So I will go in these steps one by one, and overall I will try to cover uh, obviously the introduction will have why it is required, why it is necessary, and all those things, and then we'll go for the I will put some light on the different kind of the genome sequencing technology, and how it is sequencing is happening and what kind of sequencing is necessary for that, and then we'll go for the different steps which are participating in the assembly construction part, as well as like how why we need to construct an assembly, all kind of the steps, and after construction of the assembly what we have to do, because that is also the one of the important steps we have to take care about that. And then again like in assembly, after assembly construction we have to go for different sub steps which is very important, like repeat masking, and then the gene prediction. So gene prediction also in a typical, or you can say tough steps throughout the genome, because correct gene prediction itself take much time, and it has to be validated from the different technology also. So those are the steps I'm going to focus more on. And then how we are going to go for again the pathway annotation and those kind of the things will be covered here. And then as I told, like variant calling and Jiva's part will be there. So I think I just put it in the basic information about the genome here. And what as I told, like what is genome? Genome is like a complete set of the genetic material of an organism. So it can be any kind of the organism, your microbiomes, your you can say your plant, you can say your animal, and different species of the plant. So that is like a genetic. I think everyone are aware about this. The basic terms are there. And each and every like eukaryotic and prokaryotic, all the genomes are having their all the species are having their own genomes, and these genomes are typically depends on the complexity also. So there should be like haploid genome, there should be like diploid genome, there should be like tetraploid genome. So as the genome complexity will increase, the assembly part will also, the complexity of the assembly will also increase. So I mean to say, when you have a diploid genome, it's like human is a diploid genome. So or and the identification of the human genome will take like it was started in 1990, and it take 10 years to complete that full of the human genome sequencing, and plus identification of all kind of the gene and all kind of the information from that particular genome. So see, although yeah, that factor is like that, there was no such good technology was there, sequencing technology was not there in past decade, but now we have a good sequencing technology which make this process faster. or you can say this process more faster than earlier. So now within the time frame, within 2 to 3 years, we can start a genome project and we can complete it. But again, it depends on the resources, it depends on the your quality of the genome and complexity of the genome. So like if microbes is there, it will take hardly 1 month after sequencing to complete it, because it's like virus is there, like you have heard about the SARS-CoV things are there. This is like only 30 kb genomes are there. So it depends on the size and its complexity of the genome actually. So yeah, in case of when you focus these things in the plant do have your complexity more. So there will be like diploid genome is there, tetraploid genome there, hexaploid genome is there. So definitely the complexity will increase over there, and the process of assembling those contigs, you can say, or those part will be the more take will more time, and as well as the accuracy has to be decide many things over there. And I think again, the next slide, I'm just covering the things how my genome looked like actually. So we have a DNA, then it will be wrapping up with the histone protein and forming the chromosomes, and with how you can say with the diameter of that how they are looking like, then it will go for the micromolar, and it will go for again 2 micromolar and nanomolar things. So these are the, like you can say, I just put it the microscopy over there. I find out these pictures will be more suitable and things are there. And one thing I just want to focus, after constructing the genome, we have again the complexity to find out the centroid region of the genome and telomere region of the genome. This is also very tough to get those things. So this part will be again having different technology over there, and how we have to get those sequences and all those things. And then again the question will come, why we require the genome assembly, the reason behind it. So first, yeah, definitely it's well known, we have to get the chromosomes, how many number of chromosomes are there in that particular species. So species, again I will tell the anyone, any species can be there, like your human, animal, and a different, you can say, but across the different vertebrates. Yeah, that is the one of the reason. Then again the genes which is required, necessary after getting the assembly, we have to know about the all about the genetic or genic information of that. And I will add one more point, the nowadays the non-coding part will also play the thing. So gene plus non-coding RNA is part of that genome. Then whether that particular genes are associated with any kind of the genic disorder, or you can, in case of animal also there are many disorders are coming, and in case of plants also there are many things are happening. So that is one of the things which is required. Then we can perform, after getting the genome, we can perform different comparative genomics task also. So if we have suppose in a taxonomy we have a single species over there, and we have a little information for that particular species. So when you have a new genome over there, you can have a comparative study. So this is like you can say comparing the different genomes and finding out the uniqueness of that particular genome. So we can go for the comparative genomics also. Then again, in case of human, the pharmacogenomics is one of the major part the people are doing. So although we have a different reference genome of a human, but yeah, we have to go for the like in triple IT there is like project is going on which is called like we have to sequence the across the Indian population, the genomes, and then find out the specific like personalized medicine to the Indian population. So that kind of the task will be happening, and or we can do in the after getting the genomes of the different you can say the different regional genomes population genomes actually over there, and then find out the what kind of the different variations we got in different genes over there, and those kind of a study we can go for that. Then again, after pharmacogenomics, the metabolomics is also the one of the important steps or part, you can say, are taking place, where the inside body there are different metabolites are participating and doing a lot of things in the system. Coming to the plant system, those are very important nowadays. If you are going for the Ayurveda things, and you can say there are many herbal plants are there. There, it's basically the metabolites things are participating in a more towards that. So once we have genic information, proteomics information, what kind of the reaction is going on, or what kind of the specific metabolites are produced by the particular plant, or you can say herbal plants, like suppose there are many plants are there like avla, even the mango is also having some metabolite different. So, and even the curry leaves, in curry leaf I have just worked a lot, because we have developed a chromosomal level assembly in the curry leaf, so there are specific metabolites are there, and those kind of the things we can study through the metabolites. So that's why we required the specific genome assembly, or you can say the reference level genome assembly. I will go fast here. I just put it those slides for the beginner. Those uh, don't know how the genome sequencing in an era is started, and how it is, like what is the past and what is the present, and what is like what we are going to happen in the future. So I just put it in the uh, things when we started, those kind of the things, you can say. So when it was like, it's like, you can say, history, we are recalling the history over here, and I just putting those point in a nice way so the people can recall them. And then when, so like in 1987 we got first nucleus kind of the things, which is identify the Frederick, and then again we in 900, I think there was like 8087, that was not 9087, and then 9010 we got like 5 nucleotide ways about, and then coming to the 1950 we got the patterns of the ATGC, and then discovery of the double helix in 1953, and then concept of the codon we got in 1961. Then 1968 we got the first tRNA, and then 1977 the Sanger developed the DNA sequencing first technology, which is very important point. I just want to put it there. And they started sequencing in that time, and then full genome was of the virus, that particular virus has been sequenced that time. And then a method was developed of DNA profiling in 1985, and then 1990 we well known about that human genome project was launched. And you can say I just put it the milestones of whatever happened in the past, and then first bacterium project is also started in 1995, and then 2000 we have completed the full genome of the Drosophila, and 2003 the first draft genome assembly was constructed for the human genome. It was like having 2000 to 2000 20,000 to 25,000 genes, and 2004 we have a high quality red genome, and then after 2007 we have got like multiple DNA sequencing technology which are capable to produce a million number of the genomes, million number of the base pairs over there. And then 2008 we got the 1000 genome project started over there. And then 2009 I think you people are also have to aware about that. We are doing like I just put it the base point over here. We are doing like tissue level sequencing now. In 2009 the single cell RNA, first single cell RNA sequencing was also started. So it means we are taking a particular cell and sequencing the transcriptome or genome for that particular cell, identified particular cell. So this level project was started in 2009. Then it goes in 2010 or 12, we have completed the 1000 genome project, and then 100K project was also completed in 2018. Yeah, this will be like out, you can say that 2020 or 21, it was like everything was well set up. You can, I don't say the well set up, but yeah, the technologies were well developed and things are happening. So in that way, we have multiple COVID-19 genomes are available in the different databases available. So that was the outbreak, and now I just put it the one more milestone which we got the last years, that was entire human genome decoded. Actually, decoded means we have each and every crystal clear genes available in the human genome now. So you think about that. We started in 1990, and we are working on that. And then now in 2022, the things are for the human reference genome, we got the every genes properly. So it took more than 20 plus years to get those stage. And just put it the latest picture, how these sequencing technologies are enhance the number of bases submission in the NCBI. This is particular stats from the NCBI I just took there, and how the bases as well as how the sequences are submitting throughout the genic sequencing as well as the whole genome sequence also. So you can see in the left right hand side of mine, the WGS sequencing are more happening across the after the 2002 or 2003. Before it was like gene level, people are doing the specific gene, extracting the specific gene and sequencing them. This is because of, again, the technology was not there, the things was not that proper, and then it's happening in that way. I just put here like what are the different principle. I hope these principles everyone are aware about those kind of the principle. Yeah, I just put it for the recall of those things, and I will not go in detail about this. Definitely it is well known to everyone actually. Now I'm coming to the main things, which is like why actually major steps are genome required for the genome assembly actually. And that is like we have to think, we're starting with any kind of the genome assembly project, we have to think about that, like whether that particular genome is important. First thing, if it is important, then how it is going to serve for the different you can say human kind, animal kind, and any kind of this. We have to think, because the first step itself very necessary. If it is not required right now, for it is not important for the community, or as well as it is not having a specific reason behind it, we have to think on that. If it is microbe or viruses, because those are the small genomes, we can sequence it. Now even though we are having the low cost of the sequencing also, but to like, if we have a, you can say, if we have a genome, to construct a genome, we have to spend a lot of money as well as time, as well as we have to spend the computational power over there to construct the things. So we have to clearly think about that. And then after like suppose a rare plant or species are there, so we have to think whether that species are important for that. So nowadays you can say across the India, like the people are going towards the crops. Those are like millets crops are there. Still many millets crops are not having the full genome sequence over there. So why they are important, why the people are asking about the millets, why those are important actually. So because they are having some healthy they are providing some healthy nutrients, or you can say the metabolites to the human body, That's why they are important. But how they are providing, why they are, what are the process, what are the genes are involved in that, what kind of the metabolites are coming out from there, then we have to know all about those particular information. Then we can go for the any of the rare millets kind of the things and take that project and know about it, which particular kingdom, which particular family it is coming. If similar kind of the species are different genome are established over there, then we have to think about that, whether that if a reference genome is available for that, then why we are doing this thing. So that is also the one of the concerns. So you should know everything about the genome, species by taxonomy, where it is located in that particular taxonomy, any reference genome is available or not. If not available, then we can think about that. And basically, what is the size of that genome, estimated size of that particular genome. So the genome size varies to different species to species actually. So if size is less than that, in case of rice and Arabidopsis, there is a big size differences there. Arabidopsis will have only six chromosomes and all those. In rice, we are having 12 chromosomes. The size is differing over there. So that we have to know. And then we have to go for the design the experiment workflow. So how we are going to do that, whether we are going to approach for the short read technology based, or we have to go for the long root technology based, because nowadays we have both kind of the technology which are giving us a, you can say, 99+% accurate reads over there. And so yeah, for I can put my experience over here. To construct a chromosomal level assembly, we have both, like you can say, both kind of the sequence over there, like short read also we have to require, as well as long read also we have to require over there. And then the major steps which is participating here, the library preparation part. If you are library preparation part means the extraction of the total DNA from that particular tissue of the plant, of the species, any of the species. If any library are not prepared in a proper manner, you will not get the good genome, you can say good amount of the reads over there. So this process actually take time. Time means it already the process of each technology are well defined, and each technology process are extracting from their the extraction process are well defined for the short read as well as for the long read. And then after deciding that, again in your experiment, the cost factor will also matter, and how much X sequences you are generating, that is also matter. It is recommended for, if you are generating long read base, it is recommended we have to generate at least if we have a genome size of 1 GB, we have to generate at least 10 to 20 GB of data. So then 10 to 20 GB of the data means we have to generate 20x more sequences than the actual genome are there. So in that way we have to go. And then again we are doing those kind of the things, then we have to also carefully watch our you can say, the nowadays the computational power is easily accessible when we are going for the cloud computing kind of the accessibility. But yeah, if we have a local system, or you can say the local PC or a Linux system, that genome assembly part will not complete on that. So we have to also careful about the computational power which is required for the assembling of a particular genome. And then after assembly, there are many steps are there for the checks. We have to checks. There are the checks are there. So you can say BUSCO score. We have to check whether the assembly is having the correct orientation is not. That we have to check, or are we got the draft level context over there also. Then there are many multiple steps are involved on that also. So we have to polish them. We have to again fold them, and we have to again go for the, if we got the chromosomal level assembly, then we have to again check into the wet lab whether we are having the same number of chromosome or not. So those kind of steps we have to complete. As I told, like repeat masking also important, and and then again, once we got the genome assembly, we have to submit it into the NCBI, get their certification, whether it is having the acquired parameters, or you can say the GTF genes are there, which are good or not, because genes, if you are submitting fragmented genes, that it will be also tough. They will sometimes reject them, and assembly also they will reject them. They are also having their own algorithms over there to check your assemblies. If assembly was not good, they will definitely are not going to take it, and then assembly is good, then they will take. And you can say, if any updates are coming on that particular assembly, you can submit also assembly. So that process also important when you are construction and assembly. So as you see in this particular slide, I have just given a flow chart which is telling each and every steps how we are constructing the genome, and what kind of the technology are supporting, and how we can construct it. There are like three way of construction. One is like short read based assembly generation, another one is like long read based assembly generation, third one is like you can say hybrid assembly generation. So, and people are trying the all three way, because we don't know how the things are happening inside genome, because each technology have some advantages, and sometimes they are having the some disadvantage also. So if we consider short read based, in short read will be, because we are having the short reads over there, we will find when we are assembling, sometimes we will find the gaps over there. So in an assembly, I can say there are few checks are there, special checks are there. One is like how the short reads are assembling, overlapping with each other, and after overlapping with each other, how it is going to the form a long, you can say the long DNA stretch, or your long chromosomal or long context stretch. So when I am talking about there is three things actually, which I want to make it clear. Initially, when we form the assembly, that whatever long stretch of the DNA we are getting, that we call the context. Okay. Once we again polishes or you reassemble them, then it will start forming as a sac fold. Okay. So if anyone are aware, I just want to give you an you can say exercise or work note for you people. Just try to see an assembly of any of the plant species which are not well known. So like rice, Arabidopsis, mustard, and all those things, you will not go. Or any of the other species, animal species, which are like not like buffalo is well sequenced, and just see their assembly file. You will get over there some kind. Initially, you will get the chromosomes. In some assembly, you will, wherever you are going sac fold. That means that particular assembly is not gold standard assembly. If you are getting context, that means that assembly are again the more lower stage actually. So initially, when we start assembling your DNA, your sequence reads will get the context. Then when we go for the polishing and gap filling process, we'll get the, you can say, the sac fold level assembly over there. Once the sac fold again polished by different kind of the reads, and some of the in the sac fold itself, we will get the ends over there. N means the gaps over there. To fill that particular gaps, we have to again polish them. After polishing, we may get the chromosomal level assembly. To get the chromosomal level assembly, we have to, and the number, again I told you the exercise, like we have to know about that how many possible number of the chromosomes are available on that particular species. So if we are getting the nearby number of the chromosomes plus mitochondria and chloroplast things over there, then that means your assembly are is the in the way of the gold standard. So these are the basic you can say steps of the assembly which is required for that. So uh, when we start working on that, like suppose I'm just giving one example of like here the curry leaf. We start working on that, the curry leaf, which is I have worked in this project for 3 years actually, and then we got the uh, chromosome level assembly. Soon you will get in the NCBI, and all the publications are available for that. We started with the short read based methods. We tried it, and after trying the short read based method, we are not able to get the even the nine currently basically are having 9 chromosome over there. We're not able to get the 9 sac folds. We are getting around 2000 sac folds over there after short read based technology. Then we got the data from the long reads also, and then we try to like fill gap filling of that particular long read assembly, gap filling with the long read data. Then we try when we reduced out the 2000 to 1200 something. So we thought like 1200 context is still more is there. 1200 context means these are the very fragmented assembly actually. We are not having the connectivity with the each DNA segment to another DNA, or you can say in the inside chromosome there are many fragments are there. So again, we started thinking of that, can we go with the starting with the long read, and then after long read, when we started working with the long read, and we got like luckily we got around 900 sac fold in the long read, and then we take the gap for the gap filling, we take the short read data, and then we significantly we increase the or decrease the number of the sac fold into the 100 or 200. So it was like amazing thing for us, like how it has happened. Then we start like thinking on that, can we go for the hybrid technology? So once you are not getting your objective or your number of the chromosome, you have to think about the hybrid also. So we took both the data and start polishing refinement. So it took like 1 year to doing this job actually in the computational with the good computational power. And then at the end we got like you can say the 9 chromosome plus mitochondria and all those things. So it was the things are happening. So once we have the de novo assembly, that will be, and then yeah, definitely the tools are very important over here. Which tools and which kind of the algorithm you are participating. Algorithm wise, I will go in the next slide. But yeah, there are many tools are there, and which depends on situation to situation what kind of the data are there. But I'm just putting here the specific you can say the tools, those are very frequently used in those kind of the job. So like for the long read we have two technology like Oxford Nano 4 and we have PEG Bio. So those technology are having specific tools also, so like Kanu and Falcon and Fly. These are the very known tools which are assembling the long read part. And MSET and SESTA also is a good one over there. And then when we have to generate the like you can say the small reads assembly, spread is also well known. Supernova is also good. And I will tell you this technology in the short read technology, 10x chromium technology also very important now. One more technology is that is called the high C technology. High C is basically providing you to help out the chromosomal conformation changes over there. So those like you can say that those are the short reads, but they are giving you the reads which are very rarely identified by the regular technology actually. So supernova 10x chromium technology will help to do that part, and high C part will help to do that. So the area where the regular technology will not able to extract that particular DNA or that part of the sequences of the genome, these two technology are helping to make that. So make that. And also high C is also you can say if you go with the high C detail, there are like 3C is there. High C means 3C is there. So chromosomal conformation folding is there actually. So you have to be very clear about your technology which are you after getting, because we also use the high C data. So wherever the rare gaps are there in the assembly, you have to take those input from the different technology has to be sequenced them actually. So like telomeric region, like centroid region of the chromosome, how it is assembling. So those regions we have to identify, and how the conformation of the chromosome are changing. So we have to take the help of the different technology also to get those data. Once we have those data, we have to reassemble our assembly and try to go there. So once we did that, then we then again the polishing steps I told you, the polishing steps is very important actually. So these are the different tools which are participating in the polishing steps, like Java and LORCD. Pylon is also important one, and long read for the arrow, and recon is also there. And then we go for the assessment actually. So assessment is based on the your N50 value, and also the genome coverage. Genome coverage is basically tells the how many numbers of the genes are identified on that particular entire chromosome, or you can say the draft level assembly, or you can say your chromosomal level assembly. If they are covering the all percentage of the genes, then we have to go for this. So the BUSCO is basically the major tool which is developed by the Swissport Institute, which is very important, which is telling your genome completeness, which will tell you the genome, how much percentage of the genome is your completed. So if it is more than 90% of the completeness, that means it's okay, your genome is now up to the level. But to reach to the 100% level, that will be the difficult task. Even we have constructed the genome, and we got the 97 point of the completeness. BUSCO means completeness means the genes which is identifying throughout that particular genome should have the similar kind of the tendency of the available genomes. So there are huge databases there, so we have to be matched with them. Our genome is having those sets of genome or not? Definitely 1 or 2% genes are unique for that particular species, if we are comparing to the in a particular taxa. So there are different taxas are there. So for plant, the taxa is different, or plant genes are different actually. So diploid genes will be different. Tetraploid genes will be different over there. And then once we get this particular kind of the assessment, so these kind of assessment you have to go for the multiple times. Assembly, then we have to go and check BUSCO score. If it's not falling in the criteria, if it's going less than 70%, 80%, that means that assembly is not good actually. So then we have to go for again refinement or polishing on all kind of the job actually. And then one of the more important things I just want to put it there, like as I told, HiC, and then one of the good technology nowadays is come like that is called BioNano refiner. So BioNano basically gives you I will tell you the principle of that technology. The data coming from the BioNano it's like it will give you the end-to-end information of the chromosome. So once you get your plant or any kind of the species DNA from that, you have to submit that to the BioNano sequencing process. And once we get the data, it will try to give you the optical maps for the entire genome, or maybe some of the fragmented genomes. So once you give the data, the optical maps will be generated by the BioNano. So this kind of the technology nowadays helping us to provide the faster assembly construction rate. So BioNano, once the BioNano will give you the optical maps of your entire genome, you will be able to realign with your whatever previous assembly is there and see whether the orientation of your particular genomes are correct or not. If they are not correct, it will tell you that orientation is not correct. And then we should do the realignment with bionano optical maps and try to get the exact locations, exact things over the chromosome. So this kind of the technology will help in that. So bionano aligner will also help. Bionano optical map basically give you the all the information required it. Okay. And then once you got the assembly part, then you have to go for the, once you bus score is like more than 95% and things are okay, we'll go for the repeat masking. Even though we have to also do, we can also go for the repeat masking of at contact level as well as secfold level. People are doing those part also in that level. So once we got there, because every genome will have the repeats over there. Repeats means those elements are very important actually. Lines, science elements. So these are basically participating 40% to 30 to 40% of the genome actually. So we have to go for the repeat annotation task of that. So that with the, we have to identify the repeat element. Then we have to go for the annotation. Yeah, only one king tools are there, that is called repeat masker, which is basically doing a fantastic job for doing this kind of the job actually. And yeah, the running of that particular tool is quite tricky. And in bioinformatics kind of the things, once you are going each tool details and running the job, it will be like sometimes will be messy for you, and because you are the beginner. So things have to be more precise, more clear. And when you are running the jobs, those kind of the parameters and those kind of the important things will vary to each genome to genome, or each scenario to scenario. So you will not have a setup kind of the parameter over there to do the things in a one go, like putting a sequencing to the blast, and you are getting the result. In the putting the sequencing in the blast itself, you have to define the different parameter as per the requirement actually. So my objective here to put there, each tool have their default parameter. People are going to the default parameters, but in the scenario of your own genome or your own sequences, own chromosome level things, or all kind of in your case, you have to define it. You have to do it. It will come when you are doing that exercise. So doing those particular exercise, when you are running those jobs and getting the results are differing from the previous results, and then we have to find out the differences over there, whether that particular parameters are working fine or not. So that's why I told you initially, the computational power as well as those things we have to be very carefully when we are running this. You can say computationally required jobs actually. So we have to very clear when you are submitting a polishing task, or you can say the assembly task in the computer. So that is also important actually here. And then we go for the functional annotations of the genome. So you can say after repeat, you can go for the gene prediction. And then we can create a IGB genome browser for that pure genome. And then definitely we have to do many updates and the assembly. Once, as I told initially, human genome was first sequenced in 2013, but till 2022, your entire genome, now 2022, entire genes are encoded actually. Encoded means all the information of genes are validated throughout the wet lab process actually. So in that way we have to go. So yeah, I will go in bit deep in the different algorithm which are participating in the different kind of the you can say the technology. I will not go more deep, but yeah, definitely it is like introductory of that, but a more depth will be like take more time actually. Yeah, for the short read based, we use the tools which are using they are basically like the Brueggen graphs, and for long reads we are taking the overlap layout consists, and to putting them into the correct things order. So we follow like Kamer based approaches, and Kamer based approaches, I think those are bioinformatics student. I think they have learned into the into the their maybe the classes over there, those kind of what kind of the Kamer based approaches are there, and how it is following, how the particular Kamer is forming, and how this graph approach are performing. And Kimmer is basically the word count, and when we have a read sequence over there, we have to take the word count over there, and then we, in an example, this particular example, so I just try to put that those particular words there. So we have like happy, pines, ins, and apins. So how if we are taking 4 kmer, so if we are increasing the kmer length, the complexity of the you can say a graph will increase, and if we are decreasing, sometimes there will a lot of iteration need to be participate on that, performing those Kamer based things. And here just an example I just putting over there, and and OLC, this is like older things we are using in during the times of the Sanger sequencing, to put the sequence into the one and another and find the overlaps and stretch, make the stretch over there. The same concept we have taken there. Other people has taken in the different algorithms like Fly, Kanu, and those tools we have taken, and try to make a concept of the direct graph. And once the overlap we found, and then after finding the overlaps, we go for the next to next actually. So that particular concept has been followed here. And definitely the limitation here is like computational power, and it will not perform in the short read, because it is the reads are more over there. Here in the case of that overlap technology, the reads are comparatively low with the in comparison to Illumina 1. So that is the OLC concept we have to follow. And then I'm coming to again the repeat masking task. That is like major repeats are transposable elements, lanes, and signs are there. And initially we have to find out RAP base is the database which is giving you the standard different kind of the finders are there actually. So you can say repeat, scout, piler, and LTR finder is there. They will try to find out in your entire genome. After finding them, they will go for the masking of those elements and create a particular library for that. So we have a a library of the different repeats are available. So these like repeat, scout, and repeat rep base are the library. The TIGR is also one of the library are there. Once we use those library and find out those kind of the things, so we will go for the masking of that repeats. After masking of the repeat, we have to classify those repeats. So repeat classification, or you can say the annotation, is also the one of the part over there. And once you do the annotation, that will really also, you have to see whether it is following the correct range or not. If it repeats are less over there, that means your assembly is not up to the mark. That is also one of the important things. Once you mask the repeat, then you have to go for the, you can say, the gene prediction task actually. So that is one of the important things we have to take in mind. Repeat always varies from 30 to 60%, depends on the species to species. So this is an important steps we have to follow in, basically you can say, in the plants assemblies. These are the major steps we have to follow there. Even the human and animals also, people are using this. And then again, the gene prediction methodologies. there are two kind of the gene prediction methodologies are available. One is like ab initio gene prediction, and second one is the empirical based, which is called like the reference based approaches. In empirical base, the tools are very well known tools are like you can say GeneScan, Augustus is very known well known tools actually. So you have new species, you don't know, you have a chromosome or sac code over there, you don't know what kind of the genes are available there. Augustus will give you the Augustus is the best method which will predict all kind of the genes, ab initio genes. Then definitely, yeah, we have to refine those genes isoforms. It will give you the number of isoforms over there. More isoforms are there for the particular gene, and whether genes are oriented in the proper manner or not, that we have to see. And genes are predicting from the 5' end or 3' end. That is also important actually when you are doing the gene prediction. So those kind of the information, and then going to the forming of the GTF file or GFF file of that particular gene or location, or what are the different exons are there, codons regions are there in a gene, or even the promoter region of the genes also has to be predicted in a proper way. There are the many tools are there available which are using the promoter region. You can say the whenever genes are starting, and 20 kV, 200 kV plus regions are very important for nowadays. People are looking that 200 plus region, starting from the starting of the gene, 200 kV plus part of the gene. Those are very important. They are participating a big role into the gene expression part. And I just try to make one summary over there. Those are like beginner. They have to aware about these tools, because once you are constructing a genome assembly, you should know these tools and what are their function. I think I will share this PPT to all the participant later, and then yeah, we can discuss each tools and their features, basically what kind of the feature they are having in the link over there. And feature is basically like some of the tools are based on the technology based, the technology database. Some of the common tools, they can take the input from the different technologies. So like okay, so like SASTA is running only in 1T reads, Oxford Nanopore reads, and for HagMap and Falcon is basically defined like generated by the PacBio people persons actually. And also like de novo reads is also there, magnet is there, sap de novo is there, spread is there. So like for the short read, you will get the different good and fast, because it's also we have to also think about that how fast they are working, how the algorithm is updated over there, and what kind of the error rate is there. Also we have to see the error rate of that particular tree, how much error it is giving by that particular tool actually. So yeah, although we have to try all the tool with when you are starting with the new assembly or new assembly task, and try to see which tool is giving us a good result in terms of your assembly part. And again, the checks I told, what different kind of checks are available, we have to do the check each after constructing each kind of the context. We have to go for the checks. Masurka is also good, one of the good hybrid assembler, and it has the algorithm of both like Brugian graph and OLC concept. And yeah, definitely I run this particular tool. It was giving amazing result actually when you are going for the hybrid assembler actually. So we have to try both kind of things actually. And then we have to go for the, when you have the set fold over there, then if you want to go for the you can say construct a chromosomes of those fold, Rago is working very nice actually. So that will give you the pseudo chromosomes over there. And then Raget and Ragu and Argo, these three tools are performing amazing jobs nowadays. People are using more of these tools actually. Once you have a successful level things, then you have to go for the Rago, and you have to go to the Raggeds to get the reference guiding assembly or assistant you have. So why the reference guided things are required? Suppose you have uh, from the short read you are getting thousand contexts over there, and from the long reads you are getting 200 contexts, or you can say stockfold over there. So now I want my assembly should be reference guided with the long read, and how much reads are again going and fixing to the available context actually. So this kind of the reference guided approach we have to apply. And also reference guided approach is required once you have, like suppose you have some taxonomy which is you are having the well reference genome, like in case of like citrus. So suppose citrus or is having the gold standard reference genome over there. Even the citrus till now did not have the gold standard reference, but associated species which are coming to the same family, we have to check if the same family is having the 9 chromosome. If you are in your assembly, you are getting 100 plus things over there, then we and the assembly present in the databases they are having less context, like 40 or 30 contexts are there, then we can take because those species are correlated, it has to be 9 chromosome over there. So then we can take that reference and take your assembly and guide and try to minimize the context over there. So that is the prospect of the reference guided resistance assembler, which is required nowadays to form a chromosome level assembly actually. And there are also different haplotype phase assembler is there, which is basically designed for the long read technology as well as short read. Both assembler are there, like Falcom Unichip is there, Falcom phase is there. So why the phase is required? So you have to know the phasing of the assembly also, like either they are phasing in the right direction or not, that we have to see. Some chromosome will be the when you are assembling, some chromosome will be the right direction, some chromosome will not get the correct phase of the chromosome actually. So this we have to check actually. So these are the different checker of the phasing of assembler actually. And so you can see here I just put it only the commonly tools which are participating in assembling part only. So haploid assembler is also there for the both long read and short read technology. So here the SGS means second generation technology, which is basically Illumina, and all this third generation means long read technology over there. I need I will cover two things here. One is like variant calling, another one is the JIVAS thing, because we have some time before the session close. And this is just like a overview of variant calling steps. It's not an in-depth one, because in-depth itself will take much time, and it depends on the species to species. It depends on the reference level assembly present over reference level genome quality, as well as it depends on the sequencing quality also. So once you are having the whole genome reads over there, once your people are nowadays also going for the whole exome approach also there, we can also find out the variants in the transcriptome data also there. So they're like, and if data is there, people are trying to find out the variant over there. But my main focus is that here, how to identify the variants and what is the process of identifying the variant actually. Generally, variant identification done with respect to the genomes, reference genome. Either it is you reference genome. If I'm saying the reference genome here, reference genome means it can be a sackfold level genome assembly, it can be a contact level genome assembly, it can be a chromosomal level just reference assembly. Whatever assembly is available towards that to the particular species actually. So we also have the people generally follow this steps actually. We have to generate the reads. After generating the reads, we have to go the quality check of the reads. Then we follow the pre-processing of the adapter on all those things we have followed. After the process read, we will go for the alignment with the reference. Okay. The aligner are different here. BWA, Botai 2, Novogene, GMAP. It depends on the what kind of reads you are generating. Again, here the short read or it's a long read one. So we can long read technology are the latest one, and short reads technology, you can say short read based approaches are followed by people more because of its check, check. Okay, sorry for the pause. And for alignment, here is the alignment process is important, and what kind of the different alignment we are using, that is also important. And then we'll got after that alignment, we'll get the BAM file. Okay, and this particular BAM file will go for the again processing. Processing means sorting of the BAM file and ordering of the BAM file, all those things. Then we will go for the identification of the variants. Basically, the variants is one of the best known tool is here, that is called GATK, and it do the realignment indels. It identify basically indels, SNPs. Do I need to define the SNPs or indels or inversion? Please.
Yes, sir.
Okay. Okay, so SNP means single nucleotide polymorphism. Okay. Wherever we are getting the change of a one nucleotide, we will call it as a SNPs. And how it is happening, I think there are some different mutations, things are happening across the body, and point mutations are happening, chemical mutations are happening, and then the change of a particular base can lead for the protein expression change, a protein gene expression change, all kind of the change will be happen over there. So that is happened by the one or two nucleotide change, or you can say allelic. So there is a one more term is called it's called alleles. So allele means you have 5% and a chromosome will be having your DNA is having that DNA is folding into the two direction. One is the 5' direction, another one is the 3' direction. And gene is having the two kind of the alleles information over there. So if it is in the same position, it is having TT or AT, then the allele should be the same into that. If it is one allele is changing, that may also cause the changes over there. Okay. And indels means a deletion of a certain basis. That can be the 5 bases, that can be the 4 bases, or something, or more than that. Okay, may cause the different things. Deletion sometimes stop the transcription of the protein. Deletion may any kind of the thing can be happened. So, and point mutation also will be there. So SNP may call if one SNP is SNP is changed, then maybe it will cause the stop also. It will not. It will also stop means in ORF once you have a point mutation, wherever the mutation has changed or the codon has changed from ATG to some other codon, which is referring to the stop thing. So the gene expression can be stopped over that. Like I mean to say the protein formation will be stopped. or transcript formation will be stopped. So that kind of the changes are happening. So to identify such kind of the changes, we need to determine those changes. Inversion means a gene are inverted from one direction to another direction. Okay, I hope this is clear now. Okay, so this technology will, like genetic is the one of the best tool which is well defined in the case of human. There are freebases there are SAM tool is there, and there are many other callers are there. We call them as a caller, which will take the BAM. Generally, it will take the BAM file input and try to call the uh, variants across the genome, across the species, or it variant calling can be performed in the multiple genotypes also. When I am saying the genotypes means there are multiple species data you can generate, raw data you are generating, and comparing with the reference genome and finding out the differences in the across the genotypes. So when I am saying the genotypes means number of the genic sequences has been generated. So suppose we have thousand. We have selected 1,000 people. Okay, those are some of them are having some disease. Another one having the another type of the disease, and third group we divided into the third three kind of the you can say 3 or 4 subpopulation of them, and then we try to identify what kind of the variations are happening in this particular population with respect to reference one. If we identifying some changes are happening in the some important protein, some important genes, those are responsible for uh, causing any kind of gene expression enhancement, gene or maybe the stoppage of the gene expression, or maybe it is like you can say there are many kind of the changes can be happen. So all kind of the changes can be identified. So identification of the all type of the changes across the genome in a particular sample is we called as a variant calling. Okay, throughout we are called as a variant calling. So I was in the JTK collaboration suite. After identifying the variant, we need to find out the category of them. Either they are what kind of the possible changes they are making. Okay, possible changes means are they making positive change, are they making negative change, are they making any other kind of the change. Okay, so are they making like nonsense, missense. There are the type of the mutations are there, missense mutation, and nonsense mutation. Nonsense mutation, if a base is changing but they are not changing the protein level, so that is called the nonsense mutation. One more kind of the category is there, like a start mutation. Once the you find a mutation in the start codon of that gene, that gene is not going to that transcript is not going to convert it, so that will be called as a start mutation. And a stop mutation is also there. So once we get any mutation in the stop codon, or a mutation change into the stop, a mutation causes a change and form a stop codon in between to the transcript, that is also there actually. Nowadays because of the long read technology, we got you can say we got the we can identify the inversion as well as more number of the deletion, huge number of the deletion. Huge can say the length of the deletion is can be identified. So those kind of the various variant calling we call the structural variant calling. So there is a variant calling. There is a difference between the variant calling and the structural variant calling. So structural variant calling basically large kind of the inversion is happening, large deletion is happening across the species or things. That will be basically identified through the long read technology based approaches. But for the precision medicine, or you can say the point specific medicine, you people used to follow the short read based technology methods, and they are finding the basically precise mutation. And one more thing I just want to put it over here. after finding the mutation or a variant, we have to be sure about that particular variant. Computer will give you the give you the VCF file output, or there will the mutation is there. But how we define the quality of that mutation? Whether that mutation sometimes the sequencing technology will also errorly throw that particular mutation. Okay, so what happened when we are generating the reads, we have to see how many particular reads are supporting to that particular you can say particular mutation. Suppose I am giving one example over here. Okay, this is one read is there, this is another read is there, and this is your reference sequence is there. Okay, so I know you know the basics of reads things over there. So suppose in reference we have, I'm just putting here a point, I don't know how to put the things over there. Suppose this point is A is there in a particular location, A is there. If we are sequencing 1,000 reads for this point, okay, for this location, particular location, out of 1,000, how many reads are supporting to that? So A, if it is changing to T, so how many reads are having the T? Either it is the sequencing error. Sometimes sequencing will also sequence the T over there in 1 or 2 reads or 10 reads. So we have to define the number of the quality of the variant. So we have some parameter called read depth. So we have to take care about the how many reads are supporting to the particular read. Out of 1,000, 999 are supporting. Out of 1,000, 2 is supporting. If 2 is supporting, that means that variant is caused by that things are caused, that particular mutation is caused by somehow machine error, somehow things are there. So these kind of the quality for the single point mutation as well as the large inversion, deletion, every type of the mutation we have to take care about that. So once we have the BCF, we have to go for the annotation. What kind of the things is there? I told you. So here, basically, we will discuss an overview of genome-wide association and how it is identifying what are the different tools are associated to identify those kind of the population, large scale population based study, and can it is possible to go in the, you can say, minute level, and how this technology basically you can say the technology, or you can say the methodology is evolved throughout the generation to generation, or you can say from the starting of the genome sequencing, how this technology is evolved, and what are the current state of this technology, how the people are doing this kind of the analysis, and why it is important actually. As we have discussed earlier also with one of the good student over here, to identify the significant, here the word means significant means a lot actually. Significant variants means there is a quality check, there is a confirmation, there is a things are there. So as I told in my last PPT, like how we define a variant is significant. Significant means it should qualify some important criteria. Okay, so that will, once they follow in that particular criteria, they will come as a significant variant. Okay, and how they are how the variant are calling individual for individual species. I told you, like we have a reference and we have a samples over there. We can find out that, and we can find out the variants for the across the multiple species also. Now, once we have a multiple species VCF file with us, and now how we are going to correlate with the phenotypic characteristics. Okay, the phenotypic characteristics means your suppose in case of human, you are having the body weight, you are having the body height, you are having your eye color, you are having your you can say eyebrow, and you are having your and number of the feature of other hair color is there, your how you looks like, how your eye color is there. So a number of the phenotypic features can become from an individual. Okay, this is like a visual features we are getting. Okay. On the other hand, uh, we can get the patient level information also. This technology is also using nowadays in the we can say suppose there are n number of the cancer patients are there, n number of the diabetic patients are there, n number of certain Alzheimer disease patients are there. So across the samples, across the n number of the diabetes group, across the n number of you can say any number of the disease are there and number of the population also are there, or number of the samples are there. So either those people those are having cancer do have similar tendency or do have similar kind of the variant over there, are they are falling. Then again, if you go in the cancer, what kind of the cancer they are happening, corneal cancer, breast cancer, and all those things. So these are the subcategories is again there. So for each kind of the cancer, what kind of the gene are playing important role, and what kind of the changes happen in that particular gene. So across the population, either we are finding across the population, like suppose breast cancer is there, so across the population do we have same kind of the mutation, or similar same positions mutations are occurring there or not. So if it is occurring the same position or it is qualifying or significant is there, then we have to see the correlation between that. I just told about the human things over there. In case of the plant also, we have n number of crops are there. Okay, and each crops and each species are having their own phenotypic characteristic. Phenotypic characteristic means plant height, flowering time of the plant, and how the grain or grain quality is there. And you can say the in case of the rice, what kind of long grain is there, a short grain is there, and then test quality of also grain is there. So these traits we call as a traits. Actually, phenotypic traits are associated with the genotypic changes happening in that cell. So you heard about it in crop. Someone has developed a new variety of that. How they are developing these variety? They are causing the physical mutation, or you can say the chemical mutation in the entire genome. Sorry, not in genome. In a particular, they are making the mutation, and they making the cross things over there with the some other new variety, or you can crossing over with the each variety, and they are generating the crops one generation to two generation to third generation to fourth generation, and they are seeing what kind of the phenotypic changes are coming. So such kind of the population studies across the different phenotypic traits to genomic traits, we go for the GWAS study. Okay, this you can say these kind of the studies are traditional methods, and people are using that, and they are getting the good kind of the result. And now I just put some things which we are doing currently based on the AI and machine learning based approaches. So we are thinking here the out of the box. We are working in the technology which are capable to identify the gene expression level based on the transcriptome data, based on using the different machine learning algorithm. So that is like we are thinking here the out of the box. The traditional method I am going to tell you, but there are the people those are like Google are working in such a way to identify the out of the box solution. So here we are in the Google, we are working in the things to identify the AI based approach which can be able to correlate the genotypic phenotypic characteristics based on, and they can able to predict the things in near future. So that the things is happening. But coming to this particular topic, I will go next, and why it is required. As I explained here, all the things are there. And how again how a gene is influenced, I think that is well clear now. I told a lot of time in this series, in this lecture to also deletion, duplication, inversion, substitution, and translocation. And then when the parents are meeting and something is happening, some child is a good unaffected child, someone is the affected child is there. do the parents have some disease over there, or something is happening in through the mother. So your feature is definitely depends on your parents actually. So those kind of the studies are happening. So nowadays people are doing the GWAS study to the find out the family disease also, and correlating them. So they have the n number of the generation family data, and they find out that if some families are having some rare disease which are not identifying. So in that kind of the study also the GWAS is using nowadays. Again, this is the simple, I just put it there. And how we find out, like there is a two approach, I said, a genome wide approach, another one is the candidate based approach, gene based approach. Okay. Just putting the overall overview here, how it is happening. Once we have the population-based studies data, both genotypic as well as phenotypic, we'll go for the genotyping. Genotyping again, we can go for the chip-based approach, microarray-based approach for in case of human, because we have a well-defined chips over there. We can also go for the exome sequencing-based approach. We are taking only the exomes, subgeomes. Those are participating in certain phenotypic features or protein level information only. And then we can go for the whole genome approach also. And then next step is going towards the quality check of the data of both, because phenotypic data also be standardized and genotypic data also has to be standardized. If they are not falling into the certain quality, they have to be checked again, and we have to normalize that. If some outliers are there, some you can say infinitive data, because human are putting the data over there, so sometimes they some outliers will come over there, so we have to normalize them, we have to scale them, we have to put them in a such a over that is having the quality. So suppose we have to take some pre-processing steps here, quality check as per the phenotypic data as well as genotypic data. Then when you got the both data, we have to see the next step is followed, that is called the imputation. Imputation is the main or important things over there, where we able to identify the SNPs. Those are, suppose SNP1 or SNP3 is already well known SNPs are there. But in case of SNP2 and other SNP4, SNP5, SNP6, we have a lot of changes over there. So these kind of the SNP imputation we have to do. A reference level SNPs are there, that those are not like you can say across the population. If same kind of the SNP is coming, that means the well-known SNP is there. So such kind of effect can be possible. But in the case of SNP number 2, we are having some of the people are having T, some of the people are having A, and another kind of SNPs. There we required some kind of the parameter tuning. So suppose across the 1,000 person, out of 800 we are getting G, or remaining 200 we are having C. That means that the frequency of that particular allele or particular mutation is 800. Might be that with the sequencing technology. So those kind of the imputation we have to do, and then we have to go for again the association testing. There are n numbers of methods are there for the association testing across the. Here we are giving the example of human, and across the different chromosomes, you can see the SNPs. Those are having qualifying a certain log-P value. They will be considering. So we are associating these SNPs with phenotypic characteristic actually. So that is the process of mechanism are happening. Then after evaluating this, we have to take we can take the population. Suppose African population is there, and then Asian population is there, European population there, and American population is there. So now we have to after association testing, we've got some conclusion over that these kind of the mutation are following in the African, these kind of the mutation are following in the American people. So then we go for the meta-analysis also, or we can go for the in-depth analysis of that, why it is happening in the African people, why it is happening into the European people. So then if they are well established, those features are well established, those variant are well established, then we can go for the replication also. So now the things are well established, we can push those data into the different databases. Available data, SNPDB is there. There are many databases are there. Those are having such you can say validated or defined SNPs over there. If they are having this kind of the SNPs, maybe phenotypic character they and responsible phenotypic changes are happening in that. So those kind of things. And then post as I told that those are the post GWAS study we have to perform. I told you about the genotyping. This is the replicate slide, so I just go in the next. Quality chunk control. I just keep in mind we have to remove a monomorphic variant when we are doing the quality control. Also, we have to remove the variants those are not following the Hardy-Weinberg equilibrium equation. This is such kind of the threshold equilibrium equation where we have to the many tools are giving this kind of the score. If they are not following this particular score, we have to remove them. And filtering the SNP that are missing from the functional of individual code. Like if SNP those are not coming into the particular chart, we have to remove them also. So these are the filtering we have to ensure actually. And planned case also we have to follow the similar process. Imputation I told, these are the different tools which are participating into the imputation. Impute to, Bengal, match, and these are the tools are there actually. Well-defined tools are there. How we perform the association testing? What is actually? I just putting as a not a lecture. I just want to. You should understand the question behind it. Any kind of the algorithm, any kind of the, you can say, any kind of the tool which are using this particular concept. So there will be maybe two type of the data will be there. One is linear, another one is the logistic. Linear data will be like high blood, such as height, blood pressure, body mass index. These are the linear data. If you have a phenotypic binary change such as the presence and absence, these are the logistic data actually. I just put it there. And theory and how the linear regression model will be generating for GWAS. We have Y. Y is the phenotypic changes, phenotypic values of the vector, and W alpha plus X SB and beta XB and G plus E. So this is the basic equation. Any of the tools are following this and they are taking it and they are generating that. And the model, whatever the tools are following, they are basically ML based, you can say the association based methods they are applying. Any doubt in this equation? I think annotation I have given down. So the GWAS, for GWAS, the people those are using, these are the general linear model and mixed linear model, comparison mixed linear model. BlueChip is the very known model which is used by people. And there are the Blink is also the latest one, and which is having more capability to identify the relation between the genotypes and phenotypes. So these are the methods basically used in identification the population relationship between the phenotypic traits. Generally, once we get the GWAS, we will get such kind of the plots, and we confirm these particular SNP traits are belonging to the particular things. Here, this is an example plot. In each case, or each individual will get the different kind of the plots across the chromosome. So, like suppose you can see here, my cursor is there, and here you are getting the high number of the SNPs, which is having the observed SNP frequency is large incorporated to that. So might be these SNPs are responsible for some characteristic change in the phenotype. So this is the plot basically BMI higher based SNPs. Those people are having the more higher weight. Those responsible are responsible. These are the SNPs. Those are responsible for the high weight actually. So in that way they have plotted, and then again, yeah, expected and observed luck p value we have to calculate.